Thank you, Maury, and thank you to the organizers for the invitation to present today. It's very encouraging to have attended this meeting year after year and hear the progress that's been made across a number of different platforms, and it really shows how strong this field is now and where we expect to be, and that we can make really meaningful differences with many of these technologies. Mesoblast is a cell-based medicine technology platform. We use an allogeneic mesenchymal precursor cell. We are a publicly traded company, hence the cautionary statement. We like to describe our technology as disruptive. We certainly believe that there are a number of aspects of it that are. We are a mesenchymal lineage cell platform that is derived either from immunoselected cells or which are then expanded in culture or their progeny. Do I have a pointer with this or no? Okay. Okay. I mean, it's all right. I can. The cells are located around blood vessels in all vascularized tissues, and they respond to signals that are associated with tissue damage, upon which they secrete diverse variety of biomolecules which are responsible for tissue repair and immunomodulation. The cells are then expanded so they are scalable for widespread use. They are isolated from a number of tissues. Our platform isolates the cells from the bone marrow. We use a single healthy donor, and from that donor are able to expand sufficient cells to have hundreds, possibly thousands of doses. And they have potential for commercial large-scale expansion as well via a number of proprietary manufacturing processes that are in development. There are certain immunomodulatory properties of these cells which makes them relatively immunogenic, and that's another property that, again, allows the cells to be quite scalable and used for potential commercial use. Now, for many years, we've worked primarily on a platform that is based on tissue repair. We've recently also worked on some programs that derive from the immunomodulatory characteristics of these cells. And upon inflammation, there are certain factors that are released or produced that then induce mesenchymal lineage cells to secrete certain factors that then initiate an immunomodulatory response, and that's what's shown here. The mesenchymal cells have receptors that respond to inflammatory signals, and that results in the release of these inflammatory mediators, anti-inflammatory mediators. And that function of the cells is being developed in a number of programs that are shown here on the slide. Most recently, we have reported on some results in the rheumatoid arthritis program, and I'll share that with you today. We've also previously done work and reported on diabetic kidney disease, and there are ongoing activities in biologic refractory Crohn's disease, as well as acute graft versus host disease. Our platform is broad. We have 3 programs that are in phase 3 currently. We also recently, through a licensee, had the first program approved for acute graft versus host disease in Japan using a mesenchymal stem cell. Seen here, it's called TEM-cell. And we are also in Tier 1, meaning we're moving forward with phase 3 development in the area of rheumatoid arthritis. So by way of a little further background on where the programs are, our lead program is chronic heart failure. And we've developed this program for patients who have class 2 to 4 heart failure. As we all know, heart failure is something that has a significant prevalence in this country and abroad. And additionally, the annual incident cases in this nation are about 900,000. And both the incidence and the prevalence are growing annually. And for the patients who have class 2 to 3 heart failure, there are a number of medical modalities which provide some relief from symptoms, but do not change the overall dismal course of this disease. And likewise, patients with class 3 and 4 heart failure have chronic deterioration, and their therapies offered to these patients are heart transplant and mechanical support. So there is a very clear ongoing unmet medical need in heart failure, and we hope it's being addressed by our program. Our program is supported by extensive preclinical work and a phase 2 clinical study. The phase 2 clinical program, again, we're a small company, and so we initiated a small phase 2 program, and we had certain expectations of that program, and were initially, of course, as we're all, aimed at demonstrating safety and tolerance in our programs. But we were quite pleased to see that not only were we identifying some signals of effect, but we were seeing concordance across different measures of that effect. And that's what's shown on this slide, that we saw improvements in the clinical outcomes as well as functional capacity measured by a 6-minute walk test. And we saw also evidence of remodeling in this phase 2 program. The effects on outcome were shown best with the clinical outcome of heart failure MACE, and 2 measures of that are shown on this slide. On your left-hand panel, we're showing that with the dose, a single injected dose of 150 million cells at over 36 months, the patients who received the cells had a significantly greater probability of remaining free of a first heart failure MACE event compared to the control. So there were 0 events seen in those patients over 36 months. And in the panel on the right, we look further to try to understand, are there certain subgroups within this treatment arm who may give us additional clues about where the greatest unmet need is. And what this is showing, that patients who had the most severe disease at baseline, as determined by left ventricular end systolic volume being greater than 100, had a very consistent and strong treatment effect there. So we're seeing that all of the heart failure MACE events that occurred were in the controls who had the volumes of greater than 100 at baseline. So this information was used to support going into the phase 3 program, which is currently enrolling. We've gone into this program with a number of assumptions based on our data and others. One is that patients with large baseline left ventricular and systolic volumes and advanced heart failure are at the highest risk of heart failure MACE. And so these patients have the increased likelihood of having recurrent heart failure hospitalizations. And for this group of patients, existing therapies are inadequate and present significant economic burden as well. And so in order to confirm that the dose of 150 million cells reduces heart failure MACE in patients with advanced heart failure, the ongoing trial has been designed to enrich for patients who have advanced heart failure. And that enrichment is being done based on their prior history of heart failure hospitalizations in the prior 9 months, as well as significantly elevated baseline BNP levels. And the trial's endpoint is recurrent heart failure MACE events. So now going on a bit to discuss rheumatoid and arthritis, we have an ongoing phase 2 program. We've recently reported some of the data from this program. As we all know, this is a condition that affects millions of individuals worldwide. And while there are a number of agents that are available for treatment, many patients are intolerant to treatment. And although they achieve some improvement in symptoms, the number of patients who actually achieve a remission is quite low. So if we look at remission as defined by ACR score of 70, that only occurs anywhere from 5 to 15% of patients who are on therapies with biologics. So we feel there's an unmet medical need in this space. We conducted a phase 2 trial of 48 patients. It's a randomized double-blind placebo-controlled trial. We're looking at placebos as well as 2 doses of treatment. The endpoint primarily being, of course, safety, but a number of efficacy parameters were written into this program. And first, to describe the safety again, this program is a single intravenous infusion of cells, of 300 cells of MPCs. The cardiac program was a single injection of 150 million MPCs. So we've seen in this program that infusions are well tolerated without any untoward acute infusion reactions. There are no adverse events reported during the infusions or in the post-infusion monitoring period. And overall, the safety profile is comparable between the control patients and the cell-treated patients. And just a summary of the key parameters that we've looked at so far in the study. The first 3 rows are showing the ACR responses of 20, 50, and 70%. And what we're starting to see over cell treatment compared to control patients, and particularly looking at the higher level response rates, the 50 and the 70, that the MPCs are outperforming the placebo in terms of achieving ACR responses when measured at week 12. We also looked at the measure of disease activity, which is the DAS-28 score, and look at this defined as response as well as defined as remission. And for both of these measures, the MPC treatment has outperformed the placebo as well. So we're quite encouraged by this data. We are still analyzing further data and following patients out, but believe that this is consistent with our understanding of the immunomodulatory effects of mesenchymal precursor cells, as well as the preclinical work that was done in a collagen-induced arthritis model, and is supporting our planning for phase 3 in this area. The next program I want to talk about briefly is our chronic low back pain program. Again, a condition that affects millions of individuals worldwide. And for patients who complete conservative therapy, which is rest pain medication, often epidural steroid injections and physical therapy. Further treatment options are quite limited, and therapeutic procedures, surgical procedures that are currently conducted have a variable response rate. So surgeons report that approximately 40% of patients undergoing back surgery eventually fail to have durable responses to that treatment. So we do believe there's a continuing ongoing unmet medical need in this space. And our preclinical data supported some effects on tissue repair in a preclinical sheep model. We then conducted a phase 2 study of 100 patients with at least 6 months of chronic low back pain due to degenerative disc disease. And these are patients who had not responded to conservative therapy. And they underwent a single injection of a small dose of either 6 or 18 million MPCs into the disc. The primary objective of the study was safety, and both cell-treated and placebo patients tolerated the procedures, and the event rates between the two groups were comparable. We then looked at efficacy parameters, primarily clinical efficacy measures of visual analog scale for pain as well as function. And just in the interest of time, I will tell you that on both of those measures, the cell-treated patients outperformed the control patients out to a period of 24 months. And we then used a composite that is felt to capture clinically meaningful effects in terms of pain and function. That is what is shown on this slide. On the left side, we're seeing percent of patients achieving treatment as success at 6 and 12 months, with the MPC patients outperforming the controls. And on the right panel, we're seeing those assessments at 12 and 24 months. And so this composite endpoint is the endpoint of the ongoing phase 3 trial, which will enroll approximately 360 patients and hopefully provide good evidence for us of confirmation of these findings. And so lastly, I'd just like to share with you our acute graft versus host disease program. This is a program that utilizes a mesenchymal stem cell, which is a slightly different manufacturing platform and it is a program that also utilizes repeat infusion. So the others I discussed are both single exposure. This is an induction course for a disease, acute graft versus host disease, which, as we know, is a serious and life-threatening complication of bone marrow transplantation. Our data supporting this program are derived predominantly in children from an expanded access program that was initiated many years ago and that we acquired when we acquired this platform from Osiris Therapeutics. On the left-hand panel, I'm showing that in 241 children with this condition, that we had a response rate that was 65% overall. But importantly, the top part of this curve is showing the probability of survival for responders compared to non-responders, and I think that the visual tells a story there. On the right side, we're showing that in patients who were on the clinical trial, then also analyzed with patients from EAP, but the difference being here individuals who received only steroids and did not respond to steroids, and therefore are very different from the patients on the left that receive multiple modalities. And here we're showing that the response rates are in 81% in the expanded access program and 64% in the pediatric subgroup of a randomized clinical trial. And importantly, that we're identifying that the response rate at day 28 is seen for patients whether their prior exposure was one drug or multiple. And on the right panel also showing that in this subgroup where we did have some pediatric controls, that the cells significantly treated patients significantly outperformed the control patients in this study. So I'd like to close just by briefly going through some of the manufacturing achievements. As we all know, manufacturing is quite a challenge in this field. And our platform utilizes, first of all, on the bottom of the slide, acquisition from a donor, following which we have to develop cell banks and then expand those cell banks in culture. And then the cells are formulated, cryopreserved, and available for use in clinical trials. Some of the highlights are shown in bullets above. We have manufacturing completed for all the clinical supplies for our current phase 3 programs, and we're in the process of developing those procedures and testing those procedures required for commercial transition. Specific formulations have and are being developed across different indications such that there will be product delineation that's, we think, meaningful. And we're also working on serum-free formulations as well as commercialization using 3D or bioreactor technology. And all of the work that we're doing, of course, is supported also by an extensive intellectual property platform, which is covering not only composition of matter, but also the indications, as well as complementary technologies. And I will add that the thought of some of the complementary technology and second-generation possibilities is also a very exciting aspect of this field. So I leave you with these highlights, which I hope I've shared some insight on, and thank again the organizers for the opportunity to share our company progress with you.